Hello, everyone. Thank you for joining this virtual PEGS seminar. And first of all, I would like to thank the organizers for giving us this opportunity to present today. My name is Rony Nehmé, and I am an application scientist at Creoptix. I would like to start with a few words on the company, on Creoptix. We are based in Waedenswil, just south of Zurich City in Switzerland, and we are specialized in label-free optical biosensors based on an innovative biophysical method. And we are proud to offer a very sensitive platform enabling scientists providing the scientists with the cutting edge tools for biomolecular interaction analysis. And today I will be talking about our WAVE system. This platform allows you to do kinetic characterization and helps you speeding up and accelerating your research. But before I start with the technology itself and showing you some application performed on our system, I would like to highlight the importance of kinetics. Particularly in the drug discovery field. So often in drug discovery, you would your starting point is a library of compounds or peptides, usually purified compounds or possibly unpurified, for example, unpurified peptides or unpurified compounds, crude samples that you would like to screen to find some hits for your targets. And generally, these hits can be weak binders, or potentially with fast off rates. So this is what you would like to know at the beginning to find those hits. And then you move along the kinetic trajectory, moving from hits to leads to final product and developing your drugs by improving the affinity from, let's say, millimolar or micromolar to nanomolar and potentially to picomolar range. So this is where the affinity can be informative for you, and with the risk of picking up some non-specific binders which could have similar affinity to your hits. And this is where the kinetics play a big role to understand the interaction. And for me, for example, when you derive an affinity from any other techniques that can give you only affinity, that's not enough. You are still missing very important information. And to do so, you have to understand what lies beneath. For me, the affinity is just the tip of the iceberg, and what lies beneath of the interaction are usually the kinetics. And this is what I'm showing here, where we are showing the same affinity, which is derived by, which is shown by different kinetics. So this same affinity of 100 nanomolar are reflecting an interaction that can have very, very different on rate and off rate. So this is to highlight the importance of kinetics. So our platform, so what about our technology and our platform that allows you to do real-time interaction analysis? So usually we have what we call a ligand, which is immobilized or captured onto the sensor chip. And we flow over the second partner of the interaction in a constant flow of buffer, and this is what we call an analyte. When the analyte binds its ligand, it will create a difference in mass, which reflects the difference in the changes in refractive index on the thin layer of the sensor chip, and this is what the instrument is detecting. The change in mass and the changes in refractive index will create the binding event, which is a dose response curve. Creating what we call an association curve from which you can derive your on rate. And at some point, the sample injection is stopped, but there is still a flow of buffer that creates the molecules to dissociate. And this is what we call a dissociation curve from which you can derive your off rate. Of course, there are two ways of calculating and deriving your affinity, either by plotting the response versus your concentration, and then you can get your affinity from the response from equilibrium. So this is the kinetic from equilibrium, but also more importantly, you can get your affinity from kinetics, which is a ratio between the off rate and the on rate. And this is what Creoptix has developed, both on the fluidics technology, but also on the sensor technology, and allowing you to do have high quality data for kinetic characterization. So for excellence in this field, Creoptix has developed not only the microfluidics, but also the technique itself, the biophysical techniques that's behind the wave system, which is called the grating-coupled interferometry. So this is what I would highlight today in my talk. So a no-clog microfluidics, a new design for the microfluidics that allows you to resolve very fast kinetics, but also the possibility, because there is no clog, the possibility to use any type of samples, including very crude samples. On the other hand, with high sensitivity, which is the ability of resolving kinetics at very low responses, this is owed to our technology, which is grating-coupled interferometry, which is inherently a very sensitive technique. Together with a flexible data evaluation, the innovative and objective way of analyzing and evaluating your data, you would be enabled with a very user-friendly and intuitive software. But because we are limited in time today, I will be focusing on the fluidics and the sensitivity today. So the focus of the talk today is how we redefined the microfluidics. So this is our censorship here, which is it's a no clog, it has no microvalve. And when we say there is no microvalves, which means there is no clogging, you don't have to worry about clogging. And this opens the door to the use of any type of samples, so particularly the crudest of the samples. It's a self-contained chip, and it's considered as a disposable. And it is composed of 4 channels, as you can see here, 4 different paths. So 4 channels, giving you very flexibility in terms of using the reference channel the way you want. But the design of our chip also allows a fast transition time. What we are showing here is the injection of DMSO, so moving from buffer to the sample or from sample to the buffer happens in 150 milliseconds, which is a very fast transition time, which allows you to do very fast kinetics, and we could push the boundary of redetermination up to 10 per second. So here I'm going to show you now a video, which is a cross section of the censorship here, showing you how we can achieve this fast transition time. So at the baseline, we will be drawing a buffer from the buffer inlet, and this is what I'm going to show you here now. So the buffer inlet here, this is the baseline drawn here and then exit here, and then the sample preparation will bring the sample as close as possible to the sensing area. So this valve will be open at this stage, and then in a fraction of a second we close this valve and we open this valve, allowing the sample to be injected on the measuring area. And then during dissociation we do the opposite, we close this valve and we open this valve, allowing the buffer to move from this way from the inlet to the outlet and flushing the sensing area, allowing the dissociation. So this opening and closing of the valve happens in 150 milliseconds, and the samples will be just ready here, just close by to the sensing area, allowing the determination of very fast off rates. And this is highlighted here in this example, which is a data from the Rony Nehmé Institute in Italy, where they were working on aggregating proteins like amyloid fibrils. And we could, for the first time, show the kinetics of the THT molecule, which is a small molecule, a weak binder. But classically, in classical instrument and traditionally, you would only have an affinity from this data. You would calculate the affinity at equilibrium from here, and you would derive your affinity. However, for the first time, we could now show this fast kinetics of this small molecule of THT, which is a weak binder, high micromolar. But as you can appreciate here, when we zoom in at the dissociation curve, we can derive off rates up to 10 per second. This is an off rate of 10 per second, which means it's a 0.069 second in terms of half-life, 69 milliseconds in terms of half-life. Now, also of importance on our wave system is the sensitivity. And to understand why the grating-coupled interferometry is sensitive, is highly sensitive, we have to go back to the root of the technology. This is what I'm going to show you here. You might have heard of surface plasma resonance, which is a similar technique to the waveguide interferometry, in a sense that we are sensing a refractive index change on a thin layer of the biosensor. However, what is in contrast to the SPR, in the waveguide interferometry, there is a constant flow of light. So the light travels across the sample, the measuring area, which is a bigger surface area here, about 2 square millimeter, and probing like a fiber optic, probing what's happening here, the binding events. So collecting more binding events. Here it's very localized, which gives a small signal. However, with the interferometry, you are collecting more binding events and making the waveguide interferometry inherently more sensitive than the SPR. And it is anyway, the waveguide interferometry was known well before the surface plasma resonance. And however, to make it amenable to instrumentation, it was very challenging because it was very unstable. So it was there is a lot of instability that can come from classical interferometers. And this is where Creoptix has developed this technique, now so-called grating-coupled interferometry. This is a proprietary technique. By including gratings, which are there. These are microfabricated structures that keeps the light coupled to the waveguide, and upon a binding event, the light coupled here with the waveguide will interfere with the reference beam here, the reference light, creating a phase shift in the light, a time-dependent and robust phase shift of the light, and this is what we are measuring. So the GCI is more sensitive. We have also developed the microfluidics. So this combination, this combo of sensitivity and innovative microfluidics will allow you, so this is what I'm going to talk about today, the highlight of speeding up your research by this combo of sensitivity and innovative no-clog microfluidics, which allow you an early stage kinetic characterization for crude samples. I will start first with showing you an example of this sensitivity. This is a possibility of measuring, of deriving kinetics at very low responses, very low signals, as we can see here. The molecular weight ratio between the analyte and the ligand is quite big. So it's a small molecule binding onto a big protein, 110 kilodalton protein. And as we can see, we can resolve kinetics at very low responses. This is data from Novartis in the US. As you can appreciate here, the low density protein, we can resolve data at, this is in triplicates. The red data here in red curves are the data in triplicates, and the black curves are the fitting model, a one-to-one model. And we can appreciate resolving these kinetics at below 1 unit, which can also mimic sometimes that your protein can be not very active. So your response will be also very low, and you would appreciate to distinguish noise from actual binding, and this can be done on the wave. So the early stage kinetic characterization in crude samples, I would like to start just presenting that you can do so, it's compatible. So our innovative fluidics allows you now to do any type of samples like cell supernatant, 100% plasma or serum. You can use any type of solvent, particularly harsh organic solvent like acetonitrile. You can use cell membranes, perhaps you can use unpurified material, you can use large particles like VLPs, liposomes, and of course, large binding partners, but also crude samples. This is the highlight of what you can possibly do on the WAVE system. I will start first with some peptide binders for therapeutic antibodies, and this is a collaboration with PepScan, which is a world leader peptide synthesizer. The story of this project started with, so they wanted to select some peptides and test them versus the peptide binders onto a therapeutic monoclonal antibodies. So they have a developed a phage display platform for synthesizing peptide libraries. But so the starting point here was previous attempts to find some linear peptides or even circularized peptides failed to find some good binders for this monoclonal antibody. And some with other techniques like ELISA, there was a lot of non-reproducible binding. So they would like a platform that can screen those peptides at early stage using unpurified peptides. And this is about how the story started. But first we tested the linear peptide, purified linear peptide, and here we could show that we can do both the interaction either in buffer or in 50% bovine serum. But later they have developed this CLIPS chemistry, and they could identify nanomolar CLIPS binders. This is CLIPS chemistry. If you can find more details about this CLIPS chemistry, circularized constrained peptides on PepScan website. So they could successfully identify tight binders, nanomolar binders of peptides to a therapeutic monoclonal antibody used to treat autoimmune diseases. And what we could do for them, they provided us with unpurified peptides, crude peptides, and we could do an off-rate screen. So this is an example of an off-rate screen here, where you are looking at the off-rate, but also you can also look at the response. So the response screen, where you are looking at the response that the peptide is giving when it binds to the antibody. Of course, you would have a positive control that you keep injecting to make sure that the activity is still intact, and then you can select your hits based on the response. Of course, we could see some of the hits that they are not really good behaving peptides. So because we could find, so this I'm going to show you here some off-rate screen examples where we could derive off-rates with the whole range, across the whole range of off-rates of this unpurified peptide library. So ranging from here 0.2 per second to nearly 8 per second. And of course, we know that we can push it to 10 per second. But we could also define that some of the peptides, depending on the scaffold. So here, I'm gonna go back one slide. So this is the scaffold here would have an impact on the way you are doing your assay. So we could find that some of the scaffold here are really positively charged, which means they are sticking non-specifically to the surface and creating sometimes a mismatch also in the refractive index. So this is all in all a good way of early characterization of unpurified peptide. And then of course you can take your hits and then characterize them further. Now moving to another type of crude samples is, for example, membrane proteins. I am a GPCR specialist and I worked all my life with GPCRs. And here, at the MRC Laboratory of Molecular Biology in Cambridge, and I worked on the serotonin receptor where we were aiming for structural studies. And if you are working on membrane proteins, you know how challenging these proteins are, particularly when you have to express them, liters and liters of culture, and then you spend days, if not hours, if not days, doing your purification. So it's a very lengthy purification process to get to your final product to be able to do, for example, structural studies. So it's a large scale process and it can take up to 2 days at least, taking a lot of liters of culture. And of course, you have to stabilize your receptor, find the best detergent for receptor, etc. Here, what we are showing is that you can characterize your GPCR at the early stage. So just a quick and dirty solubilization, just a quick centrifugation, using a very little, so 30 ml of cell culture for this particular GPCR that I know, which was a serotonin receptor. And here I could measure the binding of an engineered G protein. So this yellow subunit here is what we call a mini G, which is bound upon the activation of the GPCR. with small molecule agonist. With a special injection process, we can activate the receptor prior to injecting the G protein, and then we can derive the kinetic of the G protein binding onto DDM solubilized but not fully purified serotonin receptor. So this is also something that can speed up your research, particularly when you are working with challenging GPCRs. So you can have a lot of information at the early stage of your pipeline. So now to move one step forward with molecules, with biologics. So this is, I'm showing here data on COVID-19 research. So here we are collaborated with Professor Markus Seeger at the University of Zurich, where we could identify SAI-bodies binding onto the Spike RBD of the coronavirus, SARS-CoV-2. So this started with Professor Markus had a phage display library of SAI-bodies. They did it against the Spike RBD or the Spike ECD. So they could identify 57 well-behaved SAI-bodies from the phage display, and they could analyze them with ELISA. Of course, what we could do for them, not only, so we did an off-rate screen, he's not shown here, but we could identify from the off-rate screen 6 strong binders with an affinity of 20 to 180 nanomolar. This is one example of one of the tightest SAI-body binding on the ECD spike. And then, so we could, it was in agreement with the ELISA data. However, we could give them more information about the on-rate and the off-rate. And then we could characterize it further. Of course, we could imagine that you can take all these SAI-bodies, synthetic antibodies from the periplasmic. There is nothing that can speak against that because we can do very crude samples. So you don't even need to purify those SAI-bodies. But also after you have selected your hits, your strong binders, you can characterize them further. And this is what is shown here, where we are able to characterize SAI-bodies that recognize either the same epitope, which we can see here, there is no, so this is SAI-body 15 and SAI-body 45, when they are injected together, there is no more room for binding, which means they are competing each other for the same epitope. So these two SAI-bodies are recognizing the same epitope. However, here, these two SAI-bodies, they have kind of an additional signal when they are injected together, which means they are recognizing different epitopes. And we could even characterize it even further and also validate the ELISA data by doing a competition with the ACE2, because ACE2 would bind Spike, and these SAI-bodies can bind at the same epitope for ACE2 bind, so can compete ACE2 binding. This is an example here of a SAI-body that doesn't bind RBD at all. So that means so here what we are looking at, the orange curve here is the binding of the injection, the co-injection. And the red curve is the binding of ACE2 only. And the colored curve for each SAI-body will be the SAI-body only. So what we would be interested in looking at is that if the orange curve will be inhibited. So if the binding of ACE2 would be inhibited, the orange curve will be diminished. This SAI-body doesn't even bind RBD, so there is no effect on ACE2 binding onto the RBD. In this case, the SAI-body 67 does bind RBD, but it doesn't compete for ACE2 binding. These two SAI-bodies here have a partial inhibition of ACE2 binding, as we can see here. A reduction in ACE2 binding, but not by much, so it's a partial competition. And this binder here, the SAI-body 16, when we co-injected with ACE2, there is no more room for ACE2 to bind because the SAI-body has occupied the epitopes. And this has really validated the ELISA data, and sometimes it's even quicker to do it like so after you have characterized some of the interesting SAI-bodies. So it would be quicker than doing it with ELISA. Last example with biologics, with antibodies, we're using clinical data, and this is a collaboration with Professor Adriano Acuzzi at the University of Zurich, where we started here to test antibodies binding onto Spike RBD, Spike ECD, and another antigen of the SARS-CoV-2, which is the nucleocapsid protein. So as you can see here, we started just to validate the system by using an antibody, commercial antibody for each for the spike or for the nucleocapsid, but we did dilute them in 5% of plasma. Because further studies, we would like to use clinical sample, which is human plasma. So this is what we have done here. We could derive very nice kinetics using these spiked commercial antibodies in plasma. And we went further, and we took some 8 patients that contracted COVID-19, and so we did the binding responses. We could measure the binding responses of polyclonal anti-SARS-CoV-2 antibodies in inactivated samples, plasma samples from 8 patients. And so each color here is a different patient. And this is the data on the ECD, this is the data on nucleocapsid, and this is the data on the RBD. This is an ongoing study at the University of Zurich. And we can see clearly that there are differences in, let's say, the shape and curvature and the response level of each patient, which could presumably be due to different kinetics and antibody titers in each patient. So this is an ongoing study, and it's a very interesting study on COVID-19. So this is our system component, the wave core, the wave sampler, and then the wave control, the software, and this is our disposable, the WAVEchip. This is publication using the Creoptix WAVE. So we are happy that our customers now are publishing since a while now and happy using the instrument. And so you can have a look at the different papers citing the GCI data. This is what our customers say about us. You can find it on Science Select as well, what they think about us. And if you want to stay tuned, please visit our website, creoptix.com, for more events, and follow us on social media, on Twitter and on LinkedIn. And in summary, I would like to thank you, but I would like to highlight the key take-home message here, which I hope I could convince you that this platform is suitable to accelerate your research, particularly when you are developing biologics and therapeutics. You could save time. You don't need to go and do lengthy procedures for purifications or any other procedures. So you can use crude samples at the early stage of your studies. And of course, you can use the crudest of the samples, and particularly you can validate your orthogonal data, particularly your ELISA results, with confidence. And with that, I would like to thank you, and I will be able to answer your questions later. And I'm happy to take your questions. Thank you so much, and see you soon.